Bill Jones here, the head of VP of Therapies Discovery, head of IACS over at MD Anderson, has led a tremendous number of drug development projects, many of which have made it into the clinic. And someone will introduce himself a little bit more, but certainly the best person, I think, to talk about thinking at the beginning about these target product profiles and differentiation and starting with the right strategy from the outset.
All right, thanks a lot, Sarah. And hope can you hear me at the back? Okay, good. In that case, I'll go without mic. So as Sarah mentioned, I've been in drug discovery my whole career. I spent 15 years of my life working at Merck. Saw 2 projects that kind of made it all the way through to market, and several others that have been in the clinic. And then MD Anderson, I kind of run a biotech organization within the cancer center here. And we've successfully spun out a couple of companies. We've got programs in the clinic here that are partnered up with large pharma. And so I know a little bit about this, and hopefully I can kind of try and inspire you a little bit. So what I'm going to ask you today is really kind of think a little bit about kind of what these kind of target product profiles and tech differentiation mean. Why do we need this? It sounds boring, it sounds pretty dull, but it's really to help steer what you're trying to do as your companies develop here. And ultimately, it's make sure that you develop the right thing. Make sure you're actually focused here. It's to make sure that you're kind of creating something that other people are going to care about here. At the end of the day, the money follows what it does here. Patients want something because they're going to get an improved quality of life. The payers are going to pay for that improvement in quality of life here, and so the providers need to make money here. And your customers, the first person you're pitching to here is ultimately going to be a kind of a buyer, whether that be a kind of investor that's going to put money into your company, whether it's somebody you're hoping to kind of sell your project off to at a later point in time here. But ultimately, it all comes down to having a human impact. If you can really focus your product on having a human impact here, you're ultimately going to be successful here. So there's lots of, so you really need to kind of be thinking about making sure that there is a market here for your product. Or if you don't do that, you're going to really need some wealthy sugar daddy here that's going to kind of really bankroll your project here going forward. Ultimately here, kind of the insurers are going to want to kind of reduce the total cost of healthcare here. You can charge kind of whatever you like here, but it always comes back to kind of changes in the kind of what you're actually doing for patients here. How are you kind of reducing the overall healthcare burden? The fact that you can charge so much for CAR-T is because it's so effective here. So if you have a truly meaningful project here, your price point goes up here. And then there's also the whole competitive market out there as well. And you also need to think about there are no barriers to entry about your project here. If you're developing a technology platform, is that reliant on a certain piece of equipment? Are there blocking intellectual property there, or kind of are there even kind of issues in healthcare that are going to prevent a product being developed? At one point in time in my past life, we were working on something for basal cell carcinoma. The whole way that was reimbursed here, the dermatologists were actually getting charged. They were making money from the fact that they were actually kind of excising those basal cell carcinomas. So you were going head to head with that. It was kind of the ecosystem was engineered here that those dermatologists were actually kind of the ones that kind of, they had no incentive whatsoever to prescribe the therapeutic. So it kind of come from Merck, and kind of George Merck himself said like kind of medicines are for the people and the profits will follow. And that's kind of one thing that you really want to kind of ground yourself going forward. Think about the patient and then work back from the patient here. And always ask yourself here, is what you're developing here going to be truly beneficial for the patient? So really think about kind of why all of you are here today. It's probably for a number of reasons here. Is it because you're here because you are a kind of a medical oncologist, a radio oncologist here that's really thinking about kind of trying to do something truly transformative for patients? Are you a scientist that's been working on something for a decade or more and you're trying to find a home for your science? Or are you here to make money? All of these are kind of reasonable reasons to be here, and you have to think about things in a slightly different way. All of these are really, really noble goals here, and there's no right or wrong, there's no better way for any of these. But each of these has their own unique challenge. So if you're trying to solve a problem here, this is relatively easy in the sense that it gives you a road. You know where you're trying to go there, and kind of you can keep fixated on that road here. You can break down those barriers and you can move forwards relatively effectively here. You can continually kind of question and test your hypothesis as you're going forward. The real question here is, what is your actual therapeutic, your product going to look like here? Kind of, yes, you may be targeting a particular patient subset here, but what's the right way of going against it? Is it a CAR T? Is it an ADC? Is it a blocking antibody? What's the product you're actually developing here? And so really ask yourself here, kind of back to those patients, which one of those is going to be most effective? If you're trying to find a home for your science, this is a really tough road. I mean, you may have what you think is the most interesting gene or protein out there here, but what does it do? Where is it important? You may be coming at this from a lab-based perspective here, but it's kind of really, you've got to kind of find some like-minded individuals here that live the clinical reality day in, day out here, that you can pair up with and actually kind of work together here. It's very difficult just to drive a project out of a lab here unless you've got a pretty well-defined genetic hypothesis where this is a true driver here. And it may well be that you have to kind of, your first kind of project you're working on is not the one here that's ultimately going forward. And the last one here, if you're just here to make money, it's a really, really tough business to be in there, and there's a lot easier ways of doing this. So you may want to think about kind of how much you want to kind of really push against biotech, because it is tough. So what do you get? You need to be able to really be able to communicate to your investors here. On the top here, you've really got to be able to communicate what is the unmet medical need today and what is the unmet medical need going to be kind of 5 years from now. Whilst you're developing your projects, whilst you're developing your products, there's probably at least 20 other people in the world that are doing very similar things to what you're trying to do here. And so you're going head to head with them. People are going to want those projects to be invested in as well. So really think about kind of how you can tell your story there. And then kind of working back, how are you going to show your project is going to work here? And how many patients actually are out there. Oncology is getting segmented smaller and smaller and smaller chunks of a market here. You're going to need to make sure there's enough patients out there to test your hypothesis, but then ultimately here that you can actually sell it. There's going to be enough people treated across the world here that somebody can make a program. Back in my past life at Merck here, if a project wasn't predicted to make kind of about 500 million a year, Merck weren't interested in it. That was 10 years ago. And so kind of, if you're trying to pitch to a large pharma or something here, they're going to want to make kind of 9 or 10 figures for a project here. A small, medium-sized biotech here, you can really think about kind of making profits for a lot less than that. But again, you always need to remember kind of who's actually going to buy this at the end of the day. And then kind of another secondary objective here is to really think about what's special about your science here. I mean, you need to be passionate around what you're doing here. Ultimately here, once somebody kind of is convinced about where you're taking your project, they're not going to be that interested around the novelty of it. It's nice to have a kind of something that is truly unique, that is not necessarily essential. What people really care about is kind of, does it work, and then your passion. A lot of the time people will be investing a lot in you, and so you've got to come across here really thinking about kind of that your executives in your company here, you're driving, they believe in it, they trust in you, and kind of trust is a huge thing in this business here. And you've got to be able to have that interpersonal dynamic with an investor or somebody here that's actually going to ultimately buy your project here. And then again, as I said before, somebody needs to make money here from whatever you're developing here. And if you can't adequately pitch that story and kind of how somebody's going to make money here, you're going to have a hard job attracting investors. So what's it going to take to actually kind of get right the way through here? It's going to take a lot of your time. Here, realistically, what you're working on today, regardless of where it is in the discovery continuum, if it's an idea or if you've got a prototype or you've got something here that's going to kind of move into the clinic, it's still going to take you a long time. It's probably going to be kind of 3, 5, 7 years before you actually get to test your projects here in humans. It's going to take money. Realistically, for the projects that we drive in therapeutics discovery, they probably cost us somewhere between the $10 and $15 million to get them from a lab into being able to do a proof of concept trial here. I mean, you're going to need kind of that type of money. And as I was saying to some of you earlier this week, projects go wrong. So it may cost that much, but you've got to have some rainy day money here to be able to pay for things. And it's tough. It's gonna keep. You're gonna burn the candle for several nights here to trying to make a deadline. Whether you're trying to file for an IND filing here, or you're kind of going up for a major pitch to an investor here, it does take you to kind of roll up your sleeves and kind of work long hours at the time here. And it has its ups and downs, but it kind of is an exciting business to be in. And I've been here for 25 years and kind of continue to really be passionate about it. So one of the other things to think about here is kind of really keep your eyes wide open here. You will see things on your project, you will need to evolve your project, be willing to pivot here. But whilst keeping your eyes open, kind of really keep your eyes focused in what you're trying to do. It's easy to get distracted, it's easy to go down a rabbit hole, but kind of really think about what is on critical path to be able to move your projects along there here. And as I just mentioned, you need to be willing to get stuck in and kind of get stuff done. And then also kind of don't think you're going to do all of this on your own. It's fundamentally important here that you seek out people to help you along the way. There are people out here, consultants you can engage. You don't have to kind of give them up a lot here, that can really help shape your project here. I mean, you've got to understand out there kind of who you can trust and who you can't trust. I mean, what's the right consultant? You don't necessarily have to go with the first person you do. So scout around, find the best expert to pull in and lean in and help your projects going down. And then ultimately here, be stringent. I mean, you're all coming at this probably from a scientific background here. You've probably been trained as being a quantitative scientist here. Stick to those guns here. I mean, at the end of the day, you're trying to make change the lives of somebody here. And if your project is kind of is built here on eggshells, it's going to come crashing down here. So kind of, sometimes it takes, you need to kind of take a step back, kind of move back, go back to somewhere where you were, and then move forward again from a more solid foundation here. So kind of let the data drive you. If you think you're seeing something you don't actually fully understand, or you need to go deeper, yeah, you probably do need to go deeper, and it may actually lead you off in a good direction here. And on that note, be prepared to pivot. Projects do kind of meander through development here. The first project we ever took from IACS, we knew exactly where we were going to work in the clinic. We took it off in a completely different angle because we saw more opportunity over there. So it's fine to pivot here. and let everything drive you. So coming back to where I started, or it was a few minutes ago, so really kind of think about what the unmet medical needs you're trying to target here. So as you're starting to develop your project here, kind of really think about that. The playing field is moving. If you just think about what happened in the field of non-small cell lung cancer here, at the start of this century here, it was essentially a death sentence. There were very, very few therapies available out there. But you can see there has been an explosion of pointer.
It does not have
There's been an explosion of therapies that have come down through in the last kind of 2 decades. We've gone from this to something like this here, and so so for those of you that haven't seen these before, these are Kaplan-Meier plots, and they're basically showing kind of what percentage of patients, how quickly they drop off trial here. And so in the left-hand panel, patients were dying within 9 months or so. Today, about 49% of patients here have an overall survival rate of greater than 49% here. With both of the checkpoint inhibitors, that has got pushed a lot higher. So you're trying to now think about something that's going to have a major, you're looking for something here that's going to really have a major impact on overall survival if you want to take a product into the lung cancer field here. So to belabor that point, really start thinking about where things are going to be 3 to 7 years from now. You're probably going to develop something, you're probably going to get it in the clinic, you're probably going to see a clinical signal here, but somebody who's going to acquire that from you is going to be a bigger player and is going to want to know what that product's going to do for patients in 2030 here. And keep your eyes out there for competition. As mentioned, there are other people that will be working in a similar space. For example, the last year's cohort here, there was somebody that was involved in a particular cellular therapy here. There are at least 5 other projects out there around exactly the same target, working with more or less the same type of modality here that are all going into phase 1 trials here. It's fine. You'll have a slightly different clinical game plan here, and how do you differentiate from those? So what you really care about at the end of the day here is what the label is that the FDA is going to give you, or the European Medicines Agency. So every time we kind of go to the pharmacy, we get out whatever our therapeutic in there. There is always a bit of paper that you always throw in the bin here. I doubt any of you have actually bothered to take the time to read that kind of 5, 6, 7-page pamphlet there. But on each of those pamphlets that you get in your prescription, there's always a kind of an indication or usage in there. So it says what the actual medication has been approved of. Esintrest is an HIV integrase inhibitor here, and it's prescribed here. Saludra is a PARP inhibitor for the treatment here of epithelial, ovarian, and fallopian tube cancers here. Januvia is used to treat type 1 diabetes here, etc. here. This is what you care about here, because it's what the agency have approved you for. It's what the insurers will pay and reimburse for here, and this is really important. So with that in mind, you need to start thinking back. So how do you get your label here? You get your label here by running a phase 3 registrational trial against certain endpoints here. So when you come to do that, the regulatory agencies will negotiate what they want to see. Are they happy with progression-free survival? Do they want to see overall survival here? Do they want to see a particular complete response rate in leukemias here? And so that ultimately really dictates what the readout of that pivotal trial is going to be. And it's so important you get that pivotal trial right. So ultimately here, coming back to these labels here, there's always the reported outcome in these about the actual trial. What was done on that trial? What the patient population it was tested against here, and kind of how it was all anchored. So this phase 3 trial is so important in dictating kind of what's going to happen. So this is probably the most important slide I'm going to share with you today. There's two ways of thinking about this. On the left-hand side, you can start thinking from a clinic, and you can start working backwards here. Alternatively, on the right-hand side of the slide here, you can kind of let the science drive where you're going. But kind of ultimately, as just mentioned, what you care about here is that ultimate FDA label here. And that is going to depend on that phase 3 registration trial. So you can work backwards. You can say, okay, I want that to happen. Okay, so in my clinical proof of concept trial here, how am I going to know that I'm on the right path here? So at this stage, kind of, what is that early stage clinical trial going to be? Kind of here, how are you going to show that kind of it works in the right patient population here? And so knowing that, kind of moving back into the lab here, you start asking yourself here, what that preclinical trial is going to be. Are you interested in here in doing a certain set of PDX, patient-derived xenograft models here around your particular endometrial cancer, so that you can actually run that endometrial phase 3 trial here? What's really going to give you here confidence in that your program is going to truly work here? And then kind of even working back from the whole animal into the lab here, what do you actually need to see in the lab to prove it works? So trying to think kind of back here, okay, that's one way of doing it. The other way is kind of coming from a lab upwards here as you're building a program. You may have stumbled on a therapeutic here that you're interested in. How do you start building a compelling story here that kind of it works in cell culture, it works in a distinct subset of preclinical models, and so from that subset of preclinical models, you can make a jump into a focused trial in that distinct cancer type, and then from that trial, how do you actually get together with some of the kind of the med-oncs in that disease area here to say, okay, what's that registration trial going to be? What's the comparator I need? How do I show it's actually truly differentiated? So kind of the best way is to really come at this from both ways and continue asking yourself here, kind of, is my science pointing the right way? All the time thinking, okay, how am I going to get this quickly to market? We're seeing now programs, I mean, if you think about Amgen's KRAS inhibitor, that went from kind of enter the clinic to getting FDA approved in 3.5 years. I mean, that was at lightning speed. We're seeing projects now that kind of really move from here to here at lightning speed. Where that's really, really exciting is you've got a patent ticking away in the background here. You've probably filed your patent somewhere down here. Those patents have a fuse. There's only so long that you actually have kind of patent protection to sell and make money from your project. If you can go from first in human up to registration in 3 years, that essentially gives you 17 years of patent life to make a lot of money from. And so kind of speed is an important factor in this game here. So kind of really focusing around that will help you chart your course here. As mentioned before, try and stay on course here. Try and avoid distractions, wasted capital. I'm kind of really a therapeutics guy. The same type of principles hold true for kind of devices, diagnostics, and software here. The rules may change slightly, but the same principle is here. Focus on where you're trying to go there and try and kind of get there as fast as possible, and don't lose sight along the way. And then just coming back to this slide, I mean, continually ask questions as you're going along here. It may be here that you don't have the right set of data to go in direction A. You want to actually take it off in a slightly different direction here. You don't have to get locked into this down the road, but it always helps to kind of know where true north is and what you are trying to achieve along the way. Any questions on this before we get into the second piece?
Does everybody know, I just made sure it was in the links file that I shared on the Dropbox folder. How to pull up the drugs at FDA and look at those labels, because Phil was referencing the labels for approved drugs and, you know, highlighting them as a resource for thinking about clinical trials and endpoints. Has everybody looked at those before? It's in that thing. I recommend if you have known competition that you go poke around there, because it's really illuminating to think about.
Yeah.
Great talk. I have a question. You know, if you go from the clinic or the top-down approach, go from the clinic and try to think, oh, you know, this would be the indication our product needs. But that's like, I don't know, 5, 10 years down the road when you start your phase to get FDA approval. How do you make sure that during this time there's no new therapy that's being approved by the FDA for that indication and completely destroy your?
Well, I mean, so at some point in time, your company is probably going to form a scientific advisory board. So kind of it's always good. I mean, even if you're a long, long way off the clinic, to have a kind of a couple of med oncs or hem oncs on there that can keep you honest. They kind of if you're they will kind of continue to ask questions, and then as you start kind of building kind of your strategy going forward, bring some people here that kind of have domain knowledge. So if you're focused in kind of lung cancer, you go and find kind of John Hymak or kind of Guang to kind of come and join your advisory boards here. And they will kind of, and once you've got them on as advisors here, you continue like kind of pressure test dialogue with them. The other way is just to have kind of assemble a key opinion leaders, a KOL meeting or something here, pay them kind of a couple of $1,000s here for some of their time, and engage in conversation here, and kind of, it's all about kind of surrounding yourself by people to help shape where you're going here. Yeah.
So about the patent expiration. So previously I thought, you know, it's a big issue. So it's once expired, you don't make money. But as a matter of fact, even after the patent expired, I think the original company can still have arrangement with the generic company manufacturer. It's just not exclusive anymore. You can still make money.
So that you can still make money. You just can't make a ton of money here. So then again, it depends on what you've got. I mean, if you've got a classical small molecule here, that's very, very easy for the generics companies to do and sell them to make. If you've got a kind of an antibody, if you've got a cellular therapy, there are a lot more technical barriers to entry. With that said, I mean, if you think about something like Avastin now, there are several biosimilars to Avastin out there, and kind of people are trying to eat your lunch there. MDs have a habit of prescribing kind of what they know already, so kind of for them, they kind of, some people always give a script for the original agent, whereas others will prescribe the generic. So you will make some money, you're correct. All right, so moving on here and slightly different. So kind of you've got a bit of an idea of where you're trying to get there. I think the other thing you've really got to think about is to make sure that your project has got the legs to kind of go the whole way here. And so kind of, I always say if you're setting off on a program and you're building a therapeutic, aim high. Always shoot for the best possible thing you can get there here for a couple of reasons. The first is, whatever you do, your project is, the first time you make whatever your therapeutic is, day one, it's going to look the best it is. Every kind of experiment you do along the way from thereafter, it's going to pick up baggage. It's going to have a little bit of tarnishing here. You're going to say, it's got that little off-target activity, or it kind of doesn't quite have the PK or whatever I want. But if you aim here high at the onset here, it keeps yourself honest, and it really makes sure that you come along with the best thing here. And if you come along with something suboptimal here, somebody else is going to eat your lunch. I mean, somebody will come along there. So in this business, there's two types of projects. There's the first-in-class project where you have first mover advantage here. I mean, Amgen may not have the best KRAS G12C inhibitor out there here, but they got there first, people are using it, and it's going to get hard for them to displace. Somebody at some point in time will come along with something better here. And kind of the two companies here that are going to make money are the people that get there first and the people that have got the best one here. So you either need to be pretty damn quick, or you need to kind of come along with the best thing possible you can do. So kind of thinking just again kind of about what you need to do here. Think a little bit, as I mentioned before here, about anything you need to consider about kind of as your project is going to move along here. Is the patient population you're going have poor tolerance for side effects here? Is the market entrenched here, and that you're going to, if you're going into AML, you probably end up going to have to prescribing this in combination with venetoclax? Are there barriers of entry about how you actually kind of need to administer your therapeutic here? Or kind of you going after an elderly population here where they've got they don't particularly want a high pill burden here. And are there costs of market forces here, cost of goods? Are you going to need a companion diagnostic? If so, you probably need to think about developing that very much here up front. And so that's going to have some additional cost in your project. Or can you lean over onto kind of diagnostics that are already commonly used, whether it be a kind of a foundation medicine panel? Go ahead.
Yeah, so speaking of issues for targeted patient population, FDA is starting to look at or maybe ask for some pharmacogenomic, pharmacogenetic perspectives or considerations in the development process of the drug label. You know, what is your view of how widespread that's going to be for small molecule therapeutics, and particularly in oncology over the next 5 years? And is that something folks need to be worried about as they're on the precipice of going into the clinic?
I think in that time frame, and probably further, gazing into a crystal ball, 10 or 15 years here, you're probably okay. I think kind of as kind of more and more of us kind of start getting a kind of our own genomic fingerprint here, and it becomes more widespread, I think probably 2040s or something, that's going to kind of raise its ahead here. I think for kind of in the immediate lifetime, they're starting to kind of alert us that it's coming, but I don't think we need to be too worried today. Okay, so what does it really take for you to ensure you have something here that's successful here? So the first one here is just about the rigor and the robustness of the biology. I mean, at the end of the day, it's kind of how do you ensure that kind of you fully understand what the biology is doing, and it truly is a driver here about the impact of on the cancer, that it's fundamental in kind of that immune regulation here. Be really, really sure about what you're working on here. The second piece is really about making sure that you've actually got that quality therapeutic here. And it checks all the boxes that it's kind of, once you get it into humans here, it's going to be able to do what you want it to do in humans here. And the third piece here is really focused around kind of the medical need and the clinical plan. If you can get all of these three pieces aligned, it's highly likely that you're going to be able to kind of really rigorously, robustly test your hypothesis here in humans, and kind of you hope it's going to work, and there's a very good chance it's going to work. We all get surprised. Some programs, the fundamentals, there is a difference when you go into a human, and it's different from preclinical species or from a lab. We do get some surprises, but tick all these three boxes, and there's a very good chance here it's going to work. So kind of really, I think the key piece here is down at the bottom here, validation. When you're really kind of thinking about kind of moving your projects along here, really think about what's happening in the lab. Go deep around the biology, kind of understand what's happening here, use all the right model systems you can do here, get access to patient-derived material, and collaborate here. Really understand inside out what your particular kind of therapeutic is doing to disease cells or to normal cells, so those immune cells can have an impact on disease cells here. Run the right experimental systems. You don't want to just be doing kind of 1 or 2 experiments. Get yourself fixated here into just one model system, and always use OCMA AML-free cells here for absolutely everything you do. I mean, each patient is unique, each patient's disease is unique here, and so kind of at least kind of get into kind of over the lifetime of a preclinical development program, probably into a dozen or so different models here to rigorously test the biology here, and understand kind of in what particular context that your signaling pathway is important here. So again, kind of sticking with that and belaboring that a little bit more here, kind of, so what is a good target? So we always kind of, when we think about starting a new project, pull together something that's called a target CV here, and this is essentially just our roadmap about a particular program. And the first is really around the biology, the relevance of the biology for the disease process here. And kind of so looking at kind of the target from kind of with genetic interference, with kind of gain of function, loss of function studies, even with pharmacological modulation here. Are we actually kind of understanding the biological function of that target, what it's doing downstream, what are the substrates? You're going to want pharmacodynamic endpoints for your trial here. And so how much of that do we know up front? From a tractability path, do we actually think we can develop something? I mean, we're getting into the space now that we can actually start to target transcription factors better. We now clearly are starting kind of tackling some of the elephants in the room, gene-driven targets like the RAS family. But some are easier than others. It's a damn sight easier to shut down an enzyme in a GPCR than it is to kind of regulate a transcription complex or something. Again, back to that kind of that piece of a clinical development path. Do you understand that? And then think about issues here. I mean, are there closely related family members here for your particular protein target or RNA target here that could kind of give you side effects here? Mention the competition here and think about intellectual property. And so building up a roadmap of all of these items here is a key piece. And then as you decide to kick off on a program here, really think about kind of what are going to be your kind of your reality checks along the path of the program here. Just because you start a project doesn't necessarily going to mean that you're going to do that clinical trial here. How do you actually kind of be wise with spending your money here and put a set of go, no go decision points along the way there? And the one other piece I just kind of really always encourage you to do here is get your access to clinical samples. Always kind of, even from the day one of forming your company here, really start getting access to disease tissue, building those translational collaborations here, learning more and more about what you're actually doing here, and start to understand kind of the relevance of your biology in real human tissue. Okay, and so thinking about a therapeutic modality and moving on a little bit, there's no right or wrong for what you're developing. I was in with a couple of companies earlier this week, and all of you are developing something slightly different here. There's pluses and minuses with all of these here. From a peptide standpoint here, you really need to start thinking about kind of stability and kind of cell penetration here. Down the bottom end here with cellular therapy, I mean, this is clearly an emerging field, as you probably heard from Jason, or will hear from Jason. The agency are continually typing up the regulations here. It's relatively expensive in terms of you need some specific hardware to be able to do that here, and there's very tight CMC guidelines here. So you can develop all of these. All of these are now in the pharmacopoeia here, and kind of some of you are even probably at the bottom here thinking about hybrids. I mean, we're now seeing kind of even kind of RNA antibody hybrids here for target delivery here. So really think about here what you actually want to design going forward here. So the other document I'm going to be labeling is something that we call a target product profile. So this is essentially your target of what you're trying to develop here. And this is a set of kind of aspirational goals that you want to do. So coming again is this from a small molecule perspective. Similar things exist for a kind of large molecule therapeutic. So if you want to shut down an enzyme, you want to get hold of some recombinant protein here, and you want to kind of be able to show you can inhibit the function of that enzyme here. IC50 is a concentration of the molecule that you want to do. So the first question to ask is, is your therapeutic modulating the target in vitro? Once you know it can do that, you really start thinking about, okay, so getting into a cellular context, can I modulate that? So kind of, if I'm looking at the enzyme, am I shutting down that enzyme's function in the cell? Am I looking at a phosphorylation readout? Am I looking at a change in kind of product versus substrate levels here? And then is it actually having an impact on disease cells? So kind of, is shutting down that enzyme giving a kind of phenotypical response? Am I killing those cells? Am I putting those cells into cell cycle block here? And is that specific? Are you doing that only in your disease cells? Is it open? Is it not happening in non-disease cells? So starts giving yourself kind of an aim here. Typically for a kind of a small molecule therapeutic in the target engagement and phenotypical assay, you want this to be happening at double-digit nanomolar activity here. And so kind of that all relates to kind of how much drug you're ultimately giving. Then you start thinking about kind of what else it could be doing here. There are kind of 19,000 kind of other protein coding genes out there in the body. A lot of those proteins have function here. Are we doing it? Are we hitting the right selectivity here? Do we need to start thinking about other closely related family members? Do we want to inhibit those or not here? There are certain things the agency really care about here, whether it be kind of a cardiac ion channel hERG or whether it be a cytochrome P450 here. Are you inhibiting those? Because there will be safety signals if you do so. And then start thinking about, okay, you've shown your things working in the cellular system. What is it going to do here in vivo here? Does it have the right human PK characteristics so that this therapeutic is going to be circulating in a human organism here and be able to kind of shut down the target 24/7 or whatever you want to do here? And then can you start predicting what the human PK is that going to be? How much drug do you need to actually give to a human here for it to be effective? Most of us will swallow kind of 25, 200, 500 milligrams of sized pills twice a day or something. We'll tolerate that. But if somebody's asking us to kind of take kind of 6 pills 3 times a day, I mean, that you're not going to get patients complying to something like that. And then you won't get compliance. Your therapy won't be effective. And then kind of, okay, you've got the right PK now. Is it actually, will it work in a kind of in a multi-day efficacy study? If you were to run a xenograft or kind of some immune oncology study in a whole animal, are you going to actually be seeing phenotype there? And then ultimately here, kind of, is it going to be safe? Is it going to be tolerable here? So start really thinking about what you need to be able to do there. And then from this, again, going backwards here, start thinking about how you're going to be able to get there. So as you're developing products here, you really need to start thinking about kind of what's the screening funnel to enable you to get there. I mean, sticking with the example of an enzyme here, so how do you actually have a biochemical assay, a binding assay, a functional assay with recombinant protein? How do you have some form of cellular target engagement assay to show that you're actually binding to that target in a cell here, and it's having and you're directly modulating its activity? And then how do you have a disease phenotype? I mean, it's relatively easy to set these up, but again, being stringent, think about the quality of the reagents and everything that you're using. And then it also happens to have kind of orthogonal readouts here. Don't just be reliant on one assay system and one particular readout. Strange things go on. You can get artifacts in assays and everything here. So kind of it always behooves you to have a couple of different assay formats. You don't need to run them all the time, but you can have them as checkboxes. And always look for up signals. It's really easy to shut down a signal to lose for a fluorescence readout or something. It's a lot harder here to gain a signal. So if you're back to stringency, try and look on the whole for up signals. At some point in time, you're going to be developing a series of products here. You will kind of generate a large data set. You'll get a lot of prototypes here, but always kind of think about kind of the reality check here. Is my biochemical readout in a cell correlating here with my phenotypical readout here? You expect everything to match. If it doesn't match here, there's probably something going wrong with your project, or there's something you don't understand and go fully here. Just because you make kind of 300 compounds or so on a plot here, you don't know ever which one of these is going to be your drug. At the end of the day, this red square here is niraparib. It's not the most potent thing we made on the project, but it's certainly one of the better things, and it was the overall project profile of the molecule that kind of enabled us to move forward and ultimately pick that particular molecule. So most proteins have other family members here. It's always good to think about what a bit about them there. I mean, do you want them around for the ride here? So niraparib is a part 1, part 2 inhibitor. They both do similar biology here, but they don't. So it didn't hurt that we were inhibiting both of them. And in this particular case, it's actually beneficial because the inhibition of both actually makes superior efficacy here. Or are they going to give you trouble? Are they going to give you side effects along the way? Would you be better off having a lot more selective molecule here? So ask yourself what you want, and it may be that you have a first generation and a second generation of therapeutics. The first one may give you rapid proof of concept. The second one may be that best-in-class profile. And then also think about kind of what else is out there. As mentioned, there are kind of several, close to 20,000 protein encoding genes in the body here. We're not going to know what we're doing there. There are ways out there, there are pharmacology panels out there that you can test these. If you've got an antibody, there are different arrays out there that you can test the specificity of those. Always good to ask yourselves, as you're getting closer to the one agent you want to take it ahead, to do those checks and balances and understand if you're carrying any baggage here. So assuming now that you've got something here, it's working here in a vitro system, the next question is kind of, does it actually work in the whole organism? And one question a lot of people ask is kind of, what is the right model system out there to use? I mean, you can use everything from a bog standard cell line xenograft to a patient-derived xenograft to a kind of a particular mouse engineered and then a genetically engineered mouse here. All of these are good experimental systems, and there's no right or wrong. Typically, as your program starts, you start over on the left-hand side here with just kind of a simple cell line derived xenograft here. As you get over to the right, the cost, the time, the complexity of a system comes in, and a good project probably has a blend of all of these. So the key piece here is kind of really think about a model that recapitulates what you're trying to do here in the clinic here. And then kind of, you may have one workhorse model here that's kind of you use routinely for testing, and that could be the same, or it could be another one here to understand your human PK target. But you're gonna want several. You're gonna want to be building a more and more compelling data set. So for the PART program, we used a classical MDA-MB436 xenograft. We knew this particular model was BRCA1 mutant. It was relevant for the patient population that we wanted to go after because we were specifically targeting BRCA mutant patients, and we saw some pretty compelling regressions in this model. As the program started to move forward here, we started to test a series of other models here. A lot of these were patient-derived models. We went into ovarian models, we went into kind of colorectal cancer models, into prostate. This actually is kind of interesting because it starts to show kind of where the whole field has gone. The PARP inhibitors got improved for ovarian cancer and have expanded out here, and so kind of this clearly helps show that we were able to convince Merck leadership here that the clinical opportunity was a lot larger. The other thing at the bottom here is important as well, kind of use independent labs here. I mean, especially as you're starting to move ahead with your own therapeutic projects here. Getting somebody else out there to endorse what you've got, to start publishing on what you've got, to build a story, to get interest is important here. And so sharing kind of under controlled circumstances, your agent with a kind of a key opinion leader in a particular area, getting he or she to publish here is a big endorsement around what you've got. Just showing tumor shrinkage here or tumor stasis isn't really enough. I mean, you really want to understand here what is important for efficacy. And another thing I like to belabor is this whole area of pharmacological audit trail. So kind of how much drug are you administering, kind of how much drug is circulating in the periphery. Are you showing that is inhibiting kind of your particular protein target, and is it driving efficacy here? And putting these things all together, and it's important for a small molecule, a cellular therapy, you name it, it's important here. So again, back to the PARP program here, we knew that doses of 50 mg per kg twice a day or 100 mg per kg once a day were efficacious here. We were able to measure the plasma concentrations of the compound in the human body, and we knew how long the drug was circulating for. We were able to show both in peripheral blood mononuclear cells and also in tumor biopsies here that we were shutting down PARP activity, and we were able to show that was causing DNA damage here in tumor tissue, and that ultimately here we were showing regressions here. So we knew in the lab what we needed to do here. We knew our plasma concentrations. We knew how much target engagement we needed. We took these and did exactly the same thing in the human and showed in the human it worked here. So pulling it all together, kind of, you've got these documents. They will evolve as you go in, as you know more, and kind of always ask kind of yourself here, am I close to checking that, or do I need to stay in the lab for a little bit more time here? And your target product profile will evolve as you learn more about your project. And that's okay. I mean, at the end of the day, you really need to be learning from everything you do. And then also ask others here to give you a reality check. Is your target product profile right? Get other people, get some of your advisory boards here to say and help guide you where this is going. So I mentioned here that kind of your project's going to pick up warts. It's going to get worse as it goes along here. But you've just got to ask yourself here: is it fit for purpose? Is it going to be able to do the work in the clinic? If not, get back in the lab and do a little bit more. And there's no such thing about a perfect drug. I know things about some of the molecules that are approved today here. There are things out there here. Every molecule's got its imperfection here. That's okay, but just make sure it doesn't have so much damage here that somebody else is going to eat your lunch. With that note, just get it right the first time round. If it's going to take you, as mentioned, kind of 3 or 4 years to get here to the clinic, so make sure you optimize it here. And there's no right or wrong here. There are I know a program where I think the 9th molecule is the ultimate drug. I also know programs here that took 16 molecules into the clinic before, and none of those 16 ultimately got tested here. So there's no right or wrong here, but just stick to your documents, stick to your target product profile here, and make sure you've got the best thing. And then kind of in the last couple of minutes here, just really thinking about your clinical plan here. This is where you really, really get to test your ideas here. I mean, it's truly the finishing line, but it's also the most important thing in your project. This plot down at the bottom is really just meant to show that kind of as your project moves along, at some point in time, it's going to get you a declared clinical candidate. At some point in time, you're going to file your IND here. But your value inflection here really comes is once you show it works in the clinic. You can probably license a program in the preclinical setting for kind of several $1 million. You can probably kind of get kind of 10, 25 million if you license it at the stage of an IND candidate here. When you get here to clinical proof of concept, you're looking northwards of kind of $70 million to be able to license your project. So you want to be able to carry it here and kind of getting into the clinic and trying to show it works there really needs to be a milestone. So think about that clinical plan around medical need. I mean, at the end of the day, it's an experiment. I mean, you really want to kind of have going in there, what is the hypothesis for your experiment? And it needs to be well planned out here. As mentioned, do that good science. Make sure you've got the drug circulating, show you've got the pharmacodynamic readouts here. Think about stratifying your patients. Make sure you put the patients you think are most likely to respond here. And then it's relatively straightforward in oncology to think about quantitative endpoints here. Don't compromise. You've only got one shot at this, and if it doesn't work, you're going to pay, and your company is going to pay incredibly heavily for this. Number one, find a motivated investigator here that you can trust. You want your principal investigator here to be the best person possible that's going to run your trial. This person is going to make or break your project here, so take the time to find out the right person here, that they believe in what you're doing, they've got a group behind them, they can enroll in there. Try and kind of keep things relatively tight. Try and run it on as few trial sites at the onset as possible here. You want kind of whoever that's got your baby to kind of understand it fully. If you've got lots of people touching your project here, nobody's really going to feel that ownership for it. Things can go south pretty quickly. But you always need to ask yourself, can I enroll enough subjects here in a quick enough timeline? And there's a happy medium that's kind of typically have 2 or 3 trial sites here. And then harmonize the patient population here. If you know who it's responded, like kind of invest the time to find those patients and recruit those patients. In that early clinical space, if you get 1 or 2 strong responders, that's enough to give you a value inflection point of your project here. So find the best patients to treat and get them there. And so just kind of belaboring the point about eliminating as many variables here, kind of really think about what you want to see. I mean, if you can see a kind of a frank regression here, a frank regression is worth many more cases of stable disease here. Mentioned that one already. This applies less to oncology, but more to some of the other disease areas here. Kind of avoid patient-reported outcomes because they kind of certain diseases here have huge placebo rates. And then power the trial with the right number of subjects here. I mean, you really want to be able to prove in a relatively small subset. You should be able to kind of in 20 to 50 patients demonstrate if your project is going to work or not. So ultimately here it's challenging, but it's very, very beneficial here if you get it right. So hold yourself to 3 projects here, your candidate product profile, kind of what your compound looks like, and then really focus around that kind of what that clinical trial is going to be here. If you plan, you're ultimately going to be get the success here. So kind of take the time to optimize your project here and know where you're trying to go there. And with that, you can take any questions.
I will start. So, I mean, about the, I mean, this, I guess this is a challenging question for all of us. About the target targetability. For example, the KRAS, right? I mean, 10 years ago, even 5 years ago, we thought, okay, because of the molecule biology, the structure is not druggable, it's not targetable. But now we have molecule clinic. Wind part, signaling part, still, we have good molecule. So do you think this is because of the molecule itself, is because of biology, or is because of the clinical complexity? So how do we say, okay, wind signaling pathway, right now we cannot really have good molecule. After 5 years, what's the possibility we can target?
So we're at, I mean, I would argue that we're probably at kind of one of the most interesting times from a therapeutic development perspective. If you think about the explosion of technologies that have come on the scene, both in terms of the screening, how you actually identify leads for projects, strategies. I mean, covalent inhibitors have been around for a long period of time here, but now kind of with the amount of proteomics work out there for us to understand here, kind of at a kind of a subcellular level where covalent modifications happen, that's opened up the field to go in a certain direction. We're now in a stage where we have targeted protein degradation on PROTACs. We can actually kind of take out proteins here. And then kind of even kind of where we've gone now with some of the cellular therapies and kind of a number of the conjugates, the protein therapeutics, we've got new technology continues to advance, which gives us access to new protein targets and everything. So historically, kind of the targetable genome was relatively small. Today, the targetable genome is a lot wider, and that will continue to advance. The KRAS story, I mean, we've gone against kind of a low-hanging fruit. The first projects have gone out there after G12C. We're starting to see emergence of G12D inhibitors. We will start to see the emergence of G12D, NRAS, HRAS here. Things will start progressing, but I mean, long-winded way of saying that kind of you've got to kind of pair where you are in history with what you're trying to inhibit here. Yes, KRAS was undruggable there. Some KRASs are druggable now. Certainly, we will see kind of a few more kind of mutations being targetable. Will we get to the stage where there is a KRAS inhibitor for every single mutation? I doubt it. I mean, look where we are with EGFR. EGFR has been out there since, I think ilotinib was probably approved 2005. We're now on the 4th generation here. There are still subsets of patients that have kind of EGFR-driven cancers here that there's not an adequate therapy for. George.
Thank you very much, Philip. It was really very interesting. So one point, there are now all these experiments that you show take time.
Yep.
So when you start a new company, you have an interest to create a lot of experiments in the company, not in the laboratory, academia laboratory where everything is new, it takes time and people don't know perfectly to do a lot of stuff. How can we found with a small company the very good people on the market to come to our startup instead of going to big companies, to big pharma, because everything depends on the quality of 1 to people when you start a company to do experiments inside the startup, not in the laboratory.
So, I mean, a couple of things there. So, work can get done in lots of different places. From the company's perspective, you have to figure out here what is critical here. I mean, I'm coming at it from a slightly different perspective of working in 2 well-resourced organizations. What is critical to move it forward? Company needs to have anything that's on critical path under its control, or kind of you're paying somebody to do it where kind of the company is keeping kind of your eyes directly on what's happening there. There are contract research organizations. They all vary in quality here, but it's perfectly fine to send work and get others to do work. If you start working with an academic here, you've just got to be kind of to the point you make here, kind of the quality, the timelines are not gonna be aligned with the companies here, so, find a strong collaborator, establish some ground rules around expectations here, kind of, and then kind of get engaged. I mean, if you care about the science and you're talking to the bench scientist that's doing it, the quality of that work is gonna improve. In terms of the people question here, you've got to incentivize people. You've got to kind of find ways in your company here of actually kind of making them care about your project here. Either you've got to appeal to them from just the true scientific basis of why they're doing that here, make them passionate about the cause here, or incentivize them financially to do so. I mean, there are various different ways here. We're in a very difficult labour market at the moment here. COVID has kind of really shaken things up. What makes people work? I mean, it could be kind of some form of share on the upside down. It could be stock options or something. It could be a bonus for doing things well done. It could just be kind of sharing them with praise on a daily basis. I mean, there are various different ways of kind of encouraging people to step up here. And your company's got to build an identity, a culture, and encourage people to kind of to hone to that.
So can you elaborate on that point, the critical path? You know, you're, as you said, you're speaking from the perspective of well-resourced organizations. So when you see a lot of these smaller projects, how do you start to differentiate between nice to have and have to have? You know, because I think that gets lost sometimes when you're in these really early, you know, it can be difficult.
So think about what your goal is at the next inflection point. I mean, what does your, I mean, great point, Sarah. I mean, what's fundamentally important? For a lot of the time, where you're probably sitting today, you need somebody to invest in your company, in your concept here. So I've just shown things on a macro level. Putting it back here, what's the data set that you need here to convince somebody that there is a path to get to that ultimate goal here? What do you need here to show here? And it may be an early stage here that kind of it's sufficient to show kind of some cell line models in some relevant disease tissue here. Focus on getting to that near-term endpoint, but make sure that near-term endpoint is aligned with the ultimate goal here. So what's the right data package or whatever you need to get to that near-term endpoint? And it's probably a compelling vision, or it's a compelling vision and potential, and that you are the right person to do it at the onset here.
And then when you guys are triaging projects, you know, you had your really nice TPP of what you were trying to hit. You start generating all your different backbones, or maybe there might be different antibody candidates, or at what point, like, how do you, and again, this is part of the art of what you all do, but what, in your mind, how do you think about projects when you're trying to figure out, okay, we're close and we should just keep pushing on this and creating alternatives to the 10,000th molecule? Seems crazy, right? Versus, you know, maybe this isn't druggable with today's technology, and thinking about that, although that's, as you say, expanding, or maybe we should be trying a different drug class than the one we're currently trying, because we do really believe in this target, but you know, we're just not going to get there with this strategy. How do you all, as you're triaging your projects, sort of think about that? I know it's a hard question. It's an amorphous question because it's somewhat dependent on the situation, but
we always try to push our science to the edge here. I mean, the more experiments you do on the most advanced thing you've got in your project, you want to learn about it here. But one advantage of doing that is it starts to kind of raise other things you're going to have to face with down the road, but it also kind of, you're pushing it, and at some point in time, as you push your first thing along, you may say, actually, this is fit for purpose, this is good. Niraparib kind of, what was probably kind of about the kind of 200th molecule we made in the project. And I remember my boss saying to me at the time here, what's wrong with it? What's wrong with it? So we kept pushing it here. We had better things behind, but it was kind of, that project got it so far out ahead even though we have better stuff behind, it would have taken us another 3-4 months to bring that up to a comparable position. So we pushed the wrapper ahead. I think you will have an advisory board, you will have investors. I mean, it's dialogue to have with them. This is bird in hand today. We could spend kind of 6 months, we could spend kind of X $100,000 to kind of get to a comparable position. It could be something better, it may not be. What do you want to do? I mean, at the end of the day, you're going to be answerable to a board or some investors here. And you as kind of as a senior person or a founder here can make a strong recommendation, and ultimately here kind of get a few more brains in the room and kind of make a decision. I mean, we made a decision just this week to kind of push a molecule forward on one of our programs here. We know it's got a couple of flaws. It kind of, in a micromolar level, it hits a few things that we not. We've made kind of 770 other molecules that have come along since. And we've not got something that's as good as that one. So it's kind of, okay, it's time to kind of make a decision on the project. Either we push that one ahead or we shut the project down. So we're pushing ahead with bird in hand.
That was a project where you saw the flaws, you didn't quite hit all of your specs, but you felt like there was a compelling reason to push it forward that had to do with the function you were seeing.
Yeah, no, I mean, we believe in the project. We believe there's disease relevant biology going on here. Just kind of the particular protein that we're going after, its binding site is lipophilic. It means that you're going to pick up some off-target activity, and we've got it. But we've tried 770 other things subsequently, and we can't get anything better. So this is the best thing that we can practically get from here.
Questions? Oh, you're in time. I want to hit you with one more because I think you'll probably have some interesting perspective because you work in a pretty collaborative group where you guys have, you have a lot of data, and I know you guys use a lot of that data to drive your target selection to initiate new projects. But then you had that slide, which was the drug class slide, right? The pros and cons. As you're, particularly I think in the small molecule space, you have so many options available to you for different ways of putting these molecules together and targeting. How do you start with the newest, sexiest thing? Do you start with PROTACs because you believe, or do you try the simple molecules first, or how do you think about that when you're thinking about drug classes?
So think about it from a couple of perspectives. Number one is what you're trying to do. Are you trying to block an interaction? Are you trying to shut something down? How much inhibition do you need, or do you need to actually disrupt a kind of a whole protein complex here? Mm-hmm. And so that all of which are good. So ultimately it comes down to what you need to do to be most effective. To get it, not all projects can you come along with an inhibitor for, and you actually do need a degrader here. There are challenges. I mean, all right, let's remember at this moment in time here, there are no approved degraders out there. Yes, Arvinas, C4, Chimera, are now getting stuff into the clinic. It was a road, if you speak to Andy Phillips or some of the others here, it was a tough road to get there, and it's not guaranteed those things are ultimately going to get approved. I mean, it's a new therapeutic modality that we don't know that much about, so kind of, it's not guaranteed success. I mean, so what's the choice do you want to make? Do you have the right mindset here? It's not easy to develop a PROTAC that kind of you've got to optimize two pieces here. The kinetics change depends on cell type. So kind of again, trying to understand what you're trying to do there. In terms of I guess where you're going is on the biologic side of things. Do you want a blocking antibody? Do you need an ADC? Do you need a CAR here? You need to think about what you're trying to do here. Is a blocking antibody sufficient? CARs are relatively easy to develop in the lab and to move forward, and kind of we've seen a number of places within TMC quickly get them into the clinic here because they're relatively cheap to move forward. Antibody drug conjugates here, there's a lot more protein engineering here. They take longer to do, so it's a real trade-off. But really, it ultimately comes down to what you need to do.
Can I ask one more question?
Yeah.
So this is related to making go-local decision. In old days, when there is a anti-cancer drug chemo like doxorubicin, paclitaxel, platinum, it's used in the clinic before the mechanism of actions are actually figured out, right?
Yeah.
And nowadays it's different. You really need to know the target. But in a scenario where you do observe lots of anti-tumor efficacy in animal study, and it doesn't quite hit your target, maybe there is some unknown off-target effect. How do you proceed? How do you make a decision in such cases?
So again, there's no right or wrong answer here. I mean, if you're seeing compelling activity, you could push ahead here. Again, kind of pushing a project until it breaks, and you'll get it into a toxicology study, understand here if it works, and if there is a therapeutic index, and then from there you could jump ahead. At some point in time, whether it be an investor, whether somebody's going to acquire that asset, they're going to want to know what's happening here. I mean, there are a few rare examples whereby things have gone right the way through to approval, where things aren't known. Most of the time here, kind of large pharma wants to understand mechanism of action. So kind of maybe you push the program forward while you try to kind of find collaborators to understand their mechanism of action.
So okay, my question actually is related to this, but it's really good to the TPP. So let's see that you are targeting protein-protein interaction with a small molecule, which is difficult, right? Some of the protein-protein interaction, they are transient, they don't really have a strong binding. So do you really have, is it really a mindset that, okay, so your biochemical or biophysical assay has to be below 50 nanomolar or low nanomolar? Because in this case, maybe 100 nanomolar or even 200 nanomolar will be enough to show the cellular efficacy or even animal efficacy, or even community. So is it acceptable even you have 200 nanomolar with your biochemical biophysical assay in this situation? Is it acceptable for the investors or farmers to say, let's go forward?
Again, if we were to compare various different approved therapeutics, I mean, it's a range. Yeah, I mean, there are drugs out there that are in the kind of the hundreds of nanomolar range on their binding to their target. There are things out there that have cell phenotypical responses in the kind of 5 or 10 micromolar level. If you're in that situation, the chances that you've got something else going on, you've got some form of off-target activity, are high. I mean, that tells you you've got you're not making any good, strong kind of contacts with the protein here. The risk that you've got some off-targets and non-specificity going on are pretty damn high, and with that means that kind of you've probably got some unwanted pharmacology going on here. So the advantage with aiming high is that you don't bring some of that baggage along with you, or even if it comes along with you, that there's going to be a differentiator. You've got a therapeutic index. And then the other reason is kind of if you need to hit 10 micromolar kind of levels constantly, then that means you've actually probably got, unless you've got some really phenomenal human PK, you're actually going to have to give a lot of drug to be able to get those concentrations 24/7 or something here.
Well, since like metformin, for example, their IC50 is in the higher macromolar range. Okay, it's still the most widely used, one of the most widely used drugs.
Yeah, it's one of the drugs that we still don't really understand exactly how it works. So,
Right.
so,
It's also an older drug, right? So standards have changed a little bit.
so,
All right, thank you, Phil.
I think so.